So, good evening, all. We are back with our second session in clinical research. Today we are going to have the second session um, on finding gaps in herbal and nutraceutical clinical evidences. Yesterday we discussed about different types of observational clinical trials and how to design them. So over to you, Dr. Sriharan.
Yes. Hi, good evening all. Today we are going to see about finding gaps in herbal and nutraceutical clinical evidence. What are the gaps in herbal and nutraceutical clinical evidence? First of all, herbal we know that is a plant. The nutraceutical is what? Means the nutraceutical is nothing but it is a food product like a supplement. It is like a food product or supplement which like example they are in a drug form like capsule, but they are not considered as a drug. They are in a form of capsule, example like omega-3 fatty acid. Likewise they are called as a nutraceutical. We going to find what are the gaps in this research. Today we agenda finding the gaps and identifying the method how to overcome the gap, and with the live case scenery of case study. Why this is matter means the lot of herbals and nutraceutical product have market in high range. They have in market values high, but the efficacy value is very low. So far, this is very matter. And also the patients are self-medicating the drug without any clinical guidance, without any clinical setting. Example, they are taking the drugs. We are going to the first part, the herbal evidence problem. Likewise, before I'm saying herbal medicine have large popularity. It have large in marketplace, but their evidence is very low. Scenes from herbal supplement, prescription drug, function of the food, their evidence is very low, but its popularity is high range in a graph. And the regulatory. The regulatory have double standard in pharmaceutical and nutraceutical. Why I am saying means in pharmaceutical there have a rigorous evidence required. While we apply for the new drug application means there should be a regarded evidence should be submitted to the regulatory authority. But in herbal medicine there is no regardless evidence. This is one of the lack of problem in herbal medicine. They easily accept the drug due to because of its traditional use. For example, we using neem for teeth hygienic and for using for skin irritation. Likewise, we using no. Because of this traditional use itself, they claiming as a drug approval. So the regulatory have here the double standard for pharmaceutical and nutraceutical drugs. What are the key barriers? Financial, methodological, and regulatory. We will see deep on one by one. Financial barrier. Financial barrier is nothing but while we doing a normal clinical research, clinical trial in normal drug means it take around 10 crore, and the return on investment also high. By while doing in a herbal or nutraceutical product, the return on investment is very low. Why I am saying means example there is have no patent rights belong to the company. Example we are bringing product in neem means the neem have no patent rights given to any company. All the company produce the neem. They will sell the neem product. One company sell for 30 rupee, one company sell 40 rupee, one company sell for 50 rupee. People choose cheaper rate. So there is a return on investment is very low. So they are not doing more research on herbal research. And another one thing is methodological challenge. Methodological challenge is first of all botanical variability. Because example, the neem tree growing in India versus Africa, they have different geographical location, different climate, different water, different soil. Due to this condition, they produce different phytochemical profile. they have variation takes place. And also extract standardization also very low, because while we taking turmeric means the turmeric root contain 2 to 6 percentage of curcumin component, while we taking one turmeric they have 2 percentage, another turmeric they have 4 percentage. Likewise, there is no standard extraction takes place. It should be in a different manner. It also one of the challenges. And dosage uncertainty. In normal drug, we know the exact dose, we know the duration, we know when to stop, when to discontinue. But in herbal medicine, we don't know the duration, we don't know the how long we take, how dose we take, we don't know. And placebo design, in herbal product, the placebo also, they are doing only research with placebo alone, dummy. Likewise, they are called as a dummy drug. They are not doing with other kind of option. This also takes place. While they are making placebo also, they are difficult to make. Which why means the placebo is like similar to the standard drug. While we making in placebo for garlic means they have a strongest smell and taste. So the matching of placebo design is also difficult. Regulatory paradox. Before I am saying itself, the regulatory accepting weak evidence continuously. So far that the weak evidence generation is continuously in a cycle manner. So it want to change. Why the manufacturer also not give most interest in this means return on investment is very low. The manufacturer done more research on meat, they may reveal the product limitation, which will helpful for the real clinical setting. Evidence of hierarchy of problem. This is the evidence hierarchy. How long the evidence is classified. The first level classified is RCT, second level is cohort, third level is case control study, fourth level is case series, fifth level is animal and in vitro study. In this evidence of the hierarchy, the herbal and nutraceutical products are staying within 5. They are staying only in the fifth region. So most of the herbal evidence are lying in the bottom. So due to this may affect the human real world setting. Only they doing an animal study means that won't reflect to the human study. They may have different different uses may be level. So this mismatch also takes place. Another publication bias. In herbal and nutraceutical product, the researchers are publishing only the positive results. When we go to the clinical trial registry means, if you look onto the herbal product means, you will know about all detail, what's are ongoing, what's are incompleted, what's are unpublished. Most of the articles has been unpublished. They are publishing only a high effect article, which create impact. Herbal medicine is very useful. Likewise, they are creating impact. But there are a lot of unpublished articles are in our clinical studies. Common research, what are the flaws are, because while doing the herbal medicine there is a minimum sample size is very low. There also the duration of period also very short. No, they are doing only with placebo, so controlling group, other control options are also very low, poor control group. And the points are irrelevant endpoint. Here we want to know about main thing, irrelevant endpoint mean, while we doing research taking name, it give a good effect in lab value, but it not continue with the clinical outcome. They so good in lab value, but they not continue until benefit of the patient in disease. So these are the irrelevant endpoint. They're so nice in a surrogate marker, but in clinical outcome there is no effect or less effect. Kitchen sink. The kitchen sink itself, lot of particle should be stuck in the sink. They do not know which is stucking. Likewise, like we take any herbal product means there should be mixture of a lot of ingredient. So we cannot know which ingredient is effectively producing this effect, or which ingredients is failing. We do not know. So like a synergic claim, we all know about the synergic effect. One or two drugs are given simultaneously, they give effect. Likewise, they are in a herbal product. This is one of the main problem. Clinical uncertainty. Why means the limitation of the clinical data in herbal medicine lead to physician to 75 physician have a lack of knowledge about a herbal product. Due to this limitation, evidence-based medicine, lack of this evidence-based medicine. For example, the patient asking doctor, sir, doctor, shall I take neem along with metformin, or shall I take neem itself? Means there is no answer for doctor, because there is no evidence proves that with, you take only neem, you take neem along with metformin. There is no evidence. So lack of evidence lead to the clinical uncertainty. I didn't here. Next, we move to the second step. What is mean? We going to identifying the gap. How do we identify the gap? What are the method takes place, and how to success that? We are going to see about that. Starting point. First of all, we analyzing the gap. First of all, we analyzing the gap. The starting points are database. These are the three database. We want to look into three database. Literature database. In literature database, we will see the journal article publication in there. So we can see the evidence of the list from the this literature refer through the PubMed, MPACE, Cochrane Library. These are the very large public database in a literature database. And afterwards in clinical trial registry, before I'm saying itself, clinical trial registry will say about what are the trials going on, what are the trials are completed, what are the trials are uncompleted. Using this clinical trial registry itself, we will know about what are the articles is still now unpublished. We can easily identify using the clinical trial registry. And specialized resources mainly specialized for the herbal medicine like Naprolot, Hermet, natural medicine database. These are mainly for focused resources for herbal medicine. While we doing the research on herbal medicine, we look onto this to identifying the gaps. Here the steps what we going to take. First of all, we want to define the scope. Which drug we are taking? Example, ashwagandha. We are taking ashwagandha as a specific herb. Condition: generalized anxiety disorder. Outcome: it will reduce the generalized adversity scale. This is the your defined scope. Then step 2 is comprehensive search. Using this database, before I'm saying these three database, you want to go to the comprehensive search through the multiple database like PubMed, Medline. You want to look onto the search. This database helps to give the quality of evidence to support our research. And gray literature. Gray literature is nothing but it is also like evidence. It is in the form of conference abstract or report. Likewise, it takes place. Then after you comprehensive search, you want to map the evidence like category. What are the evidence are done in RCT? What are the evidence are done in cohort? What are the things are done in cross-sectional? You want to mapping them. After mapping them, you can easily identifying the missing element using PICO format. PICO format is nothing but you will see upcoming slide. Using this PICO format, we can easily identify what the element is missing. Example, I'm saying one population. Well, the study done only with adult means we easily know the elder patient and pediatric patient is missing. Likewise, we identifying the missing element. After identifying this, all the gap is not important. Some gap is unimportant. So we want to prioritize the gap, which is high important, which is low important, which is moderate important. Likewise we want to prioritize the gaps, and we go to the research. PICO format. PICO format is very useful for gap identification. Population. Likewise, we are doing a research in only in adult, like NEEM studies with adult under the diabetes 2 mellitus means we easily know here the under not representative patients are elder population and pediatric. In intervention, in intervention, we can identify the gap using the preparation method. Likewise, if the study takes place in neem, only the study takes place alone in the neem extract. They omit the neem dry powder, they omit the neem tincture, they omit the neem oil. Likewise, we want to identifying the gap. And comparatory, the study only takes place in placebo alone. There is no other clinical outcome takes place. There is no other control group is included. Likewise, we want to comparatively identify in the gap. Then outcome, the outcome, some of the outcomes should be ignored by the researcher. We want to identify that outcome. What are the outcome can be ignored, what are the outcome can be omitted by the researcher, we can identify through the outcome. PubMed search. The PubMed search is nothing but, first of all, step one is you want to make a keyword. You for identifying the keyword using this PICO format, you can identify the keyword. Example, population. Here itself, the elder population missing. Likewise, these four from you identifying the keyword. The four of them you you will lead to identifying the keyword. After identifying the keyword, and the second thing is you want to search using this PubMed search strategy. MES is nothing but medical subject terms. It is a medical subject term. But how do we search how do we search in PubMed research? Means there is a Boolean search type, and or and not. And means your searching should be in a broad way, and or means your searching should be in a narrow way, and not means you will excluding the study. Likewise, example, if you taking the herb name ashwagandha and disease is generalized anxiety disorder in clinical trial, means it will filter out the irrelevant data should be avoided. Using this method, irrelevant data should be filtered out, relevant data should be shown. It will very useful for quality of to make the quality of evidence. Likewise, the PubMed search strategy is very useful for doing if you are doing any systematic review on any research, means PubMed search is very useful. And if you're doing a meta-analysis, means PubMed search is very useful. So PubMed search strategy is very useful. While we identifying the gap, follow these two steps. Step one, first of all, we identifying the keyword using the above PICO frame. And next thing is search under the Boolean characteristic and or not. Clinicaltrial.com advanced search. This is the one of the database run by the government. In this database, you can able to know about what are the research are going on, what are the research are completed, what are the research are registered, what are the research are still now unpublished. Using their intervention condition status, we can identify all those things. This is very useful. This is the evidence map template. While identifying the evidence, you want to create one type of template. This template very much useful to what is missing and what should be included using this evidence map template. So this evidence map template consists of study design, population, PICO format, population, intervention, duration, outcome, and quality. Which type of quality it should be have. And in this template itself, we can easily identifying the missing evidence. So this template is very important to identifying the gap in research. Afterwards, we know about the normal pyramid. The inverter pyramid is so very and before itself, you see the inverter pyramid very low is in vitro and animal study. Likewise, this in vitro in animal study lead to the evidence gap. Why I'm saying means why I'm saying means this only takes place in animal study, not in any human research or takes place. So this inverter pyramid assessment is very much important. Another one is qualitative gap assessment. Using this, we can assessment the gap assessment. What means? Firstly, itself we know there is no human trial. The lack of human trial takes place, and another thing is only tested in healthy population. So there is no uses of it. Why means we can do one either with not disease status patient alone. It can be very useful. And another thing is short-term outcome. It only short duration of the studies alone take place. There is no long-term effect is take. And another thing is there is no comparison group. Firstly, I'm saying itself there is no comparison. The studying only takes place in placebo alone. So there is no lack of comparison group. This is a specialized database, NAPRAT. NAPRAT is nothing but natural product alert database. In this database, there have 60K plus plant with ethnobotanical and biological activity data. Ethnobotanical is nothing but the study of relationship between plants and human. How they using the sources. Example, neem. Neem is a plant. How the people using that? These uses, botanical uses are using for the dental hygienic, using for the bacteria, any skin irritation. Likewise, this is the botanical uses. Cochrane review. Cochrane review is one of the standard reviewer which evaluate the medical intervention effectively. Why I am saying means the Cochrane review have some golden standard, because while the reviewing means these golden standards should be met. What are they are means that should be unbiased, and their methodological should be transparency. Their methodological should be transparency, and they should follow the strict inclusion and exclusion criteria. After these criteria met alone, the Cochrane reviewer should be review the article and publish their research. While in herbal medicine, they are difficult to meet these criteria. Why I am saying means there is a lack of evidence. Due to this, they difficult to meet the criteria in Cochrane review. So they only have 135 review in herbal medicine, and most of the review, Cochrane review itself declined. This is insufficient evidence. 60% of the herbal review they declare as a insufficient evidence in conclusion. Gray literature assessment. Gray literature assessment is not like a commercial published article. It is like a report publishing in a conference abstract, or thesis presentation, or industry releasing their white paper about this any research, on government releasing any report regarding to regulatory. Likewise, this is a gray literature assessment. This is also have quality of evidence to support the research. While we searching, this is two type. We want to first identify the targeted site, and next one is file type. Targeted site, if you want go with abstract conference means type abstract site conference abstract, file type PDF means it's automatically come, and it will be very useful for identifying the gap in literature, identifying gap in research. Likewise, thesis. If you have any related assessment you want to search, means you go with thesis. Targeted site is thesis, file type is PDF or report or anything you want to search in Google. It will give the result for this. It also one of the literature, one of the database to assessment for gap. And another one is indigenous knowledge. Indigenous knowledge is nothing but disconnection. Disconnection. Why I am saying disconnection, the word means in our country or in our anywhere, the herbal medicine have, we know about his uses, preparation and method, but all in the form of oral or in the local language or in the community record alone. There is no, that record should be published in a global research database or scientific research database. This disconnection also lead to gap. So these are the things should be avoided. Likewise, I am saying the traditional use database rarely linked to scientific literature. This is the point. I'm saying that the traditional use database are rarely linked to the scientific literature. Use of this lack of local language and oral form due to disconnect in the herbal research to in a global and scientific database. This is the safety data. This safety data, the light yellow shows that individual evidence gap between all those things. We will see from the upcoming slide. Research disparity analysis. The research disparities analysis, why they coming means the 90% of the clinical trials are done in only 10 countries alone. In our world, only 10 countries alone. All these 10 countries are well economic country. And traditional use related drugs are only in the middle income country or low income country. So they have no sign in clinical research. This is also one of the drawback. Before we see the safety data gap, this is by following the first one. Herbal drug interaction gap. In herbal drug interaction gap, in clinical study, animal study, they have shown these are the drug can cause the herbal drug interaction. So only in the animal study, but it's not reflected in the human study. So the lack of herb drug interaction in human studies takes place. And second thing is only done in only in vitro studies alone, lab test alone. So it does not correlated with the clinical relevance. So it is in still now unclear manner. Another one is pharmacovigilance. In drug, we have reporting center, we have monitoring, but in her drug interactions are unrepresentative and unmonitoring. These are the main gap in safety concern. Quantifying missing evidence. Till date, there are 50 selling top 50 selling drug in current our international global herbs. These herbs are 73% they have lack of RCT. They're only done by only animal study, in vitro study, lack of RCT, and they have no placebo control or related study. Only they study with the placebo effect. They have no comparative effect test. They still know they are unclear, and duration also very less. And those finding studies, extract standardization also still have lack of 91% have lack of dose finding study. Till now they have no clarity on that. These are the quantifying missing data current selling herbs in international level. Today, now we going to see about the case study in neem diabetes. We all know the neem biological name is Asadecia indica. It is using under the 2000 years ago as a traditional use in Ayurveda. The primary claims are it used to reduce the blood glucose and the insulin sanitization. What are the active compounds that take place in Asadecia indica are nimbin, quercetin, acidica. These are the active compounds that takes place in neem. Here the neem diabetic evidence pyramid gaps. Here we see it only there is 2 RCT till now and 0 cohort study and 3 case serious study takes place. There is much larger in 4th and 5th. Animal study is 15, in vitro studies are 26. The study itself states that there should be a lack in human trial. Why means they are only in the bottom level itself. So this is the main evidence showing the gap in the clinical research. What are the key limitations while doing the neem RCT means there is a minimum sample size. Likewise, before I'm saying minimum sample size and duration also very low. They are only in 12 weeks. That is not enough for doing any. If you're doing with any chronic disease, mean 12-week duration is not enough, and they have limited endpoint. Whether the surrogate marker is not correlated with the clinical outcome. So limited endpoint and poor reporting. And during the study, there any adverse effect or undesirable effect takes place means that also not reporting. Due to the system, they are also not reporting. They have poor reporting system. Here the neem clinical trial data with animal data. This yellow, light yellow showing the trial data and dark brown showing the animal data. You itself see there is larger in animal data, it's the data only. There is lack of human data. Neem preparation, massive variation. Likewise, I before say methodological variation and extract standardization differentiation. Likewise, leaf have a different form of preparation. They have in a extract manner, they have in a powder manner, they have in a oil manner. Likewise, but all these have no standard extract. They have different extract. Due to this different extract, the result should be in a various. This result should be seen to the variation. So it also not to attain exact dose. What exact dose should be given? Whether the dose should be stopped? How the toxic dose? Due to this lack of variation, we cannot attain the actual dose, minimum dose, and toxicity dose. This is the pie chart of the neem dosing gap. Here itself saying that the dosing is automatically it's claimed only by using traditional use alone and animal exploration. There is lack of human pilot study and proper dose finding. These silver saying that they're using the drug because of their traditional use. They use in ancient period. Likewise, they following. There is no event or evidence supporting that. Likewise, if you're taking in Ayurveda hospital, they take you take this neem for daily. They won't say how many gram, how many days. Duration is lack of duration takes place and lack of dose. These are the things that lead to the gap in herbal medicine. Neem safety gap analysis. Likewise, there is no data available in drug-drug interaction with metformin, and if in CKD patient or renal failure patient means renal and hepatic impairment is mainly consideration. So it is also lack, and safety in pregnancy also we don't know, and it also not used in study like long term, only in 12 week. It is not enough for doing any acute chronic study. The neem evidence gap patient population. Here missing, likewise before I am saying that the population is missing. Sometimes they include adult alone. They are missing elder and pediatric patient and age gap. Age gap. Likewise, there are the elder patient, pediatric patients are not in this study. There are that the age gap also takes place and comorbidity. They only including the studies like if we taking the leave, they only study outcome like diabetes alone. Like no comorbid takes place. Likewise, hypertension, CKD, the comorbidity condition also not taken in the care while doing the research. So these are the kind things also lead to the evidence gap in neem research. Comparative effectiveness gap. Likewise, there is no head-to-head trial. This head-to-head trial, lack of this head-to-head trial, which will reflect lack of clinical real setting. Why means we only done in placebo drug means the real drug effect is unknown. So we cannot implement this in the real clinical setting. So that while patient, likewise before I'm saying patient asking means while we take metformin alone or along with neem means doctor have no evidence to answer their question. So this is the main effectiveness gap, comparative effectiveness gap in neem. This is the research pipeline. Till now, what's going on in neem? There are only four ongoing in a preclinical, and phase 1 they have a one pharmacokinetic study, and phase 2 two small efficacy trial study, and phase 3 there is zero. This is from the clinical trial government official website. Part 4, case study on wheat grass in cancer. It is also one of the herbal product. We are going to see about the marketing and marketing evidence and actual evidence. Their marketing evidence, they saying they fight for cancer, they're reducing the chemo side effect, they boost the immune function, they detoxifying the body. Likewise, they claiming their marketing for advertisement purpose, but reality, the reality in which they take place in only in vitro study alone. There is no randomized control study. It also takes place in a small manner alone, only with 220% alone. And mechanism also clear. Whether the effect of the wheatgrass also is still now unclear. Whether what is the actual effect, clear effect is unclear. And there is no evidence of this is there is no evidence of detoxing evidence. They are detoxifying the body. Till now, there is no current evidence from any published article from the database. Till now, there is no evidence supporting them. Here, the current evidence of the wheatgrass in cancer. Like it start from 2007. 60% takes place in randomized clinical trial for chemotherapy-induced neutropenia. In this study, that so the wheatgrass juice or in any form that reduced the neutropenia level in cancer patient. They show some effect, but due to this small population, the wheatgrass in cancer cannot shine as much. So lack of population gap, it will not shine likewise. And 2000 level, the study takes place. This study takes place only in animal cell. cell study showing apoptosis in leukemia cell. Oh, no, no, the study takes place in human cell. These study only 20 percentage, 20% has been requirement this study. That is not enough. And they also show while we taking the wheat gas in a juice form, it will reduce the apoptosis leukemia cell in a cellular formation stage itself, it reducing, and it also show from the some of the patient in the study. And 2015 it is a in vitro study takes place in a mouse. It also under the induced cancer induced mice, using cancer induced mice. These also show reducing the tumor growth in while taking the wheatgrass. It also revealed that reducing the tumor growth in mice. And pastly the current 2019 a 20% pilot study takes place for chemotherapy induced nausea. While we doing a chemotherapy, the major side effect is nausea and vomiting. While we taking the wheatgrass, it also reducing the effect of the nausea and vomiting, and it also uh, it also clinically observed from the patient while doing the study in 2019. Wheatgrass component mismatch. Likewise, their phytochemical profile is mismatched. Why means in lab study they test purification test takes place. So they extract the chlorophyll from the wheatgrass. But in commercial product, they take the whole wheatgrass. They're taking the juice. So there is the difference and mismatch between the wheatgrass component in the clinical lab value and real human setting. Here the safety gap in patients are microbial risk. While we taking the fresh juice of wheatgrass, it also causes some microbial risk, which will be give risk for immunosuppression cancer patient. So it have a microbial risk. And nutrition impact. Due to this chelation property, while we taking the narrow therapeutic drugs, it will affect the drug absorption. So it also some impact in nutrient and drug-drug interaction. Still now there is no data related to drug-drug interaction, and adverse drug event also till now there is no data related to this. What is missing in a critical wheatgrass gap? Till now, there is no survival rate is published, and how much the quality of life assessment, how much patients should be gained from this. Till now, there is no evidence on that, and still now there is no claim. Wheatgrass claim the prevention of cancer. Still now, nobody claim. So these are the missing element in the wheatgrass. This is the graph shown in the wheatgrass. What are the wheatgrass trails should be registered, and what are the trail is not registered, what are the trail is unpublished. The first is showing that registered, and the second is showing that completed. And after completion, till now there is a lot of unpublished article takes place in wheatgrass. This graph indicating that. Then we go to the final part is practical gap analysis and framework. How should we practically analyze the gap using some framework? This is the card template. While we analyzing the missing element or gap, we want to create a template like this, and identifying the five major thing. Firstly, whether the evidence is clinical efficacy, it takes place in long-term safety, and mechanism validation is whether we know the theory of the effect, which drug is causing, whether the theory of effect is well known, and dose optimization. Extract standardization is known or unknown, and it also used for special population like elder patient or pregnancy patient or pediatric patient. We want to take all these evidence domains as a first thing, and after comparing our result, we will give the mark like strong, limited, missing. What are the things are strong, you will put the tick mark on that, and what are the things are missing, what are the things are limited. Following by above two slide, you above two table, you put the tick mark. 5 steps to herbal gap analysis process. While we doing the herbal gap analysis, you want to follow the 5 step. First of all, you want to search comprehensive search. Likewise, before I'm saying the from you multiple database, you want to search the comprehensive. and categorizing the evidence. Likewise, the study design, quality, relevance, you want to characterize using PICO format. You want to characterize which study they take place, whether the intervention group, what are the intervention group are included, what are not included, what are the comparative groups are tested, not tested, and what are the outcome or ignore, omitted, what are the outcome or ignore. Likewise, you want to identify, categorize them and identifying the gap. After identifying, you want to prioritize. Likewise, all the gap is not important. What your study need, that gap you take as of a high priority as a first, and followed by moderate and followed by a limited. You want to take place. After this prioritization recommendation, prioritize, you will make a recommendation from this answer. We make recommendation. Well, we take a strong prioritize recommendation as a first and do the research and identifying the gap in herbal research. What are the critical questions while we checking the gap analysis? We want to identifying these checklists is main important. Whether there is agreement on the active compound, is whether the drug have the effect. The theory of effect is known. While we do not know means we cannot attain come to the conclusion. So first of all, we know the theory of mechanisms that we first we know whether the drug how the effect and how it creating, whether in a single way, other way, other in a mixture of the way, we want to first of all identify. After identifying, we want to look onto the standardization, whether this drug have a standardization, quality, consistency takes place, and whether it should be in a optimal dose, and whether it should be in a initial dose. Likewise, we want to standardize the thing. And after this thing, while we doing the lot of herbal drugs have animal data alone. So that the animal data supporting the human finding, whether the animal data supporting the human finding in a real clinical setting, we want to identify. After identifying that, whether the clinical should be relevant to their outcome. Why means the likewise lab values show effect in the study, but that not continue to the outcome. They only show in lab valuation itself, surrogate itself. They not come with the outcome of the disease. So they lack in lab valuation. Whether we want to identify that. This is the regulatory decision support tool. This support tool, here the light yellow indicate minimum evidence, and dark brown indicating the evidence of herbal medicine. So in regulatory, taking only the minimum level of evidence, they giving approved drug for the herbal product, which lead to lack of evidence and claiming effect in the global range, and lack of dosing effect. These are the regulatory drawback while they choosing their drug in a normal claim itself, normal traditional use itself. They are claiming their approve. They are giving approve. This is not followed by upcoming section. Why means this lead to the lack of efficacy in the herbal product, and also lead to the large gap in a study. From gap to research, when we identify all these gap, which will give the good research opportunity for all the researchers. So identifying the gap is very much important, which will be give the pay wave to the researchers to grant great opportunity while doing an herbal product or nutraceutical product. Identifying the gap is very much important. Evidence-based policy implication. These are the things could be implicant. Regulatory reform should be changed. Why means the regulatory should be changed his reform. They approving only in the minimum level of evidence. So this should be changed. And research funding should be given for the herbal evidence, which will help me to identify, reveal the limitation of the product, and it also create a post-marketing surveillance for the herbal product also. Now there is largely in drug herbal product. We want, while we creating the herbal product, we also know about the what are the adverse drug reaction, what are the undesirable event. We can easily get detail from applying the post-marketing surveillance. Patient communication framework. The patient communication framework is very helpful for the clinician researchers also. Why I'm saying means this is a data, like a data collection form. If you giving any drug, it is very helpful for giving evidence herbal based medicine. We know the evidence based medicine. This give the way for the evidence herbal based medicine. While we taking any drug, what is the level of the support should be identified. Whether it should be strongly support, it should be moderate support, it is limited support. We want to identify. And what are the unknown include? If safety should be lack. Whether safety is whether in good manner or lack. Whether efficacy is good manner or lack. Whether the interaction or takes place or not. Likewise, we want to make one framework which will help the clinician to take as a decision making tool for future. Takeaway, these are the takeaway. You can, after you go through this, you can identify in the what are the evidence landscape takes place and how to assessment the gap analysis, and what are the things behind the publication data, what are the lack of publication, unpublication, what are the literature should be available while we searching. These are the takeaway things you achieve through this seminar. Next step is, after you go through this all the slide, you apply this on the framework and do anything related to herbal. If you doing any in simple way, I'm saying means if you're doing any systematic research means you go with systematic research. Go to PubMed. Take using MES, you create a relevant article. Take the relevant article. Do the systematic review on that and publish that, which will give the gain the publication for you, publication for your resume also, and it also gives the evidence for the upcoming future review. Thank you all.
Thank you, Dr. Sriharan. It was nice presentation. So we'll just take a couple of questions are there. So from Harshal, how do difference in global regulation affect the quality of clinical evidences?
Why means the different country following the different regulatory. So till now the regulatory have approved the drug uses of the traditional uses. They do not ask demanding or asking any level of evidence supporting this herbal medicine. Likewise, they not demanding. So this is the lack of regulation takes place while we doing clinical evidence.
Okay, thanks. Next question is from Bhavesh. What is the evidence for the safety and efficacy of this specific product or ingredient in modern clinical trial compared to traditional use?
Why means this evidence is very much important. In traditional use, there is no support of any evidence. There is no, they are all in a form of oral form. They are otherwise in a local language, otherwise in unpublished in a community record. So these while we doing research or doing any evidence gap analyzing, these will helpful to identify the safety and efficacy gas in product, which will also give the way to the doing model clinical research in clinical trial.
Okay, thank you. There's one more question. So how can modern approaches, biomarker, network pharmacology, or AI-based trial designing help to bridge the evidence gap?
Yes, previously I'm
So that
saying that these last class decentralized trial. These are the decentralized modern approach. While we doing a research on a cohort or observational study means these technique is very much useful. Using a biomarker like watches or app, which one create while we taking the name. Likewise, ashwagandha related to the herbal product means using this approach we can easily fill the gap while publishing the result on the PubMed or any other Cochrane research article. We can help to identify the lack in the evidence.
Okay. Thanks. Uh, Thank you, Dr. Sriharan. It was nice presentation.
Thank you, sir. Thank you for the
Simultaneously,
opportunity.
you helped in clarifying the questions from the audience. So thank you. Thank you for joining. Thank you all
Thank you.
who have joined and contributed in the form of the questions. Thank you.